Once that message exits through the nuclear pore, it's now floating around in the cytoplasm. And inside the cytoplasm are the ribosomes that can come along and interpret the message that the nucleus sent out. Here's one large piece of a ribosome, and here's the small piece of a ribosome. And together they make up the chef. And here, as you can see, there's that chef reading that RNA recipe. And here is what this looks like actually inside of our cells. Here's the ribosome, and here's the message that it's reading. Now, ribosomes aren't just readers, they are also catalysts. That means they speed up chemical reactions by lowering activation energy. So if we make a little graph right here, we've got reactants here and products here. And the activation energy that's normally required to make those products is this much. When the ribosome is around, you don't need quite as much activation energy, and you can more easily and quickly get to those products. Now, what are the reactants and the products in this case? Well, ribosomes speed up the attachment of amino acids into their polymers, proteins. So, in other words, ribosomes are catalysts for amino acids to turn into proteins. And here's a picture of that happening in action. So now back to this diagram. We've got the ribosome in the back here, and it's acting as a catalyst, and it's acting for the catalyst for the construction of a protein. But it's constructing this protein in a very specific way. It's reading the message that's inside the messenger RNA 3 letters at a time in order to determine which amino acids to add into that protein. And to do that, it needs help from one last molecule called tRNA. The T stands for transfer because the tRNA transfers amino acids from their tip onto the forming protein. So you can see this tRNA coming in and it's delivering the next amino acid to be added to this protein as the ribosome reads its mRNA. tRNAs are perfect for helping our ribosomes read mRNA because they have letters that are complementary to the letters on the mRNA. So let's say the ribosome gets along to this triplet of A's right here. In order to know which amino acid to add onto the protein, the tRNA helps. The tRNA with UUU on it has just the right amino acid on its tip, so that when it comes and hydrogen bonds with these 3 letters, the ribosome can grab that amino acid and add it onto the growing protein. So the tRNA transfers an amino acid onto the growing protein and helps the ribosome to speed up that process of building proteins. To continue the old analogy, it's almost as if the tRNA is a shopper, picking up food for the chef to use in their meal and delivering it. And once the ribosome has attached 2 amino acids together, an old tRNA can leave and make room for a new tRNA to come and enter. And so, as the ribosome moves along the mRNA, it reads 3 letters at a time, each time adding another amino acid. Until eventually, it gets to the end, and here you can see a protein that is almost fully formed, 1 amino acid joined onto the next, joined onto the next, and the very last tRNA is just about to leave. Now this begs a question: how is it that our genes, in other words our DNA, make sure that just the perfect order of amino acids ends up inside the protein? Remember, inside our cell, there is a nucleus, and inside that nucleus is a bunch of DNA, the original recipe book. And a page out of that book is copied to make mRNA. This process, transcription, is the first step. So the code inside our genes right here gets copied into an mRNA message. That mRNA message gets sent out of the nucleus into the cytoplasm. Where that message is read. The 2 parts of the ribosome latch onto the mRNA, and then tRNAs with the right amino acid on the end come to the ribosome and allow them to start building a chain. And the further the ribosome gets, the more amino acids get added on to this growing protein. That's translation, going from this mRNA message to this protein product. Whenever a ribosome gets to a certain 3 letters on the mRNA, it always recruits the same tRNA, bringing the exact same amino acid. That allows scientists to generate charts like this, where in the center of this chart, you can see all of the nucleotides that would make up these 3-letter sequences inside the mRNA. And then on the outside of the chart, you can see the names of the amino acids that might end up inside. Take, for example, leucine. If in the mRNA there was a C, and then a U, and then a C, that would recruit the appropriate tRNA, bringing a leucine amino acid on the end of it. So CUC inside the mRNA means that leucine is going to end up inside the protein, and that's how you get from the sequence of nucleotides in the mRNA to a translated order of amino acids inside the protein. So let's take an example. Let's say that above here is the sequence that is occurring inside the DNA molecule. In order to figure out what the protein looks like, we need to do steps just like what the cell does. So first off, we need to transcribe. That means imagining what letter would hydrogen bond or come across from this C. What letter would come across from this A and this C and so on. From our knowledge of DNA and RNA being a very similar molecule, we know that across from AC comes AG, and across from an A, in DNA comes a T, but in RNA comes a U, and across from a C comes another G, and I can continue in this fashion. Then we got to do the second step just like the cell does it. We got to translate that RNA into the protein sequence. And to do that, we can consult our handy-dandy chart. Since the first 3 letters in this RNA sequence are GUG, I can look on the chart starting from the middle, G, U, G, and I know that the first amino acid that's going to end up inside the protein is valine, val for short. And we can continue in that manner. So let's see. UCU would be UCU, that's serine. So is UCA. Next is phenylalanine. And then last, let's see, UGA. Oh, that's kind of funny. UGA says stop. Stop codons like those 3 letters, UGA, signal to the ribosome to stop translating because it has reached the end of the protein. So at that point, the ribosome will disassemble and release the protein, and the protein, now fully formed, can float around in the cytoplasm and do its protein thing, and cause you to look a particular way. To review, we now know how we get from our genes to our traits. It happens through a series of steps. The first step is transcription, in which the sequence of nucleotides in DNA is turned into a new sequence, a complementary sequence, of mRNA nucleotides. Same language, so transcription. Then that mRNA is sent out of the nucleus of the cell to the ribosomes, where it is translated into a new language, the language of proteins. That language is made up of series of amino acids, and the order of those amino acids make specific proteins, and those proteins with their specific amino acids give us our own specific and unique traits.